Welcome, everyone. Welcome to Straight Science. Straight Science is an evening seminar series put on by University of Alaska Fairbanks, UAF, Northwest Campus here in Nome, and also UAF Alaska Sea Grant of Nome. This is the work office, and both UAF Northwest Campus and Alaska Sea Grant, we are public servants of the Bering Strait region, which is the waters and homeland of the Inupiaq, St. Lawrence Island Yupik, and Central Yupik peoples. And tonight, who doesn't want to hear about ice seals on a fine fall day? Tonight we have three speakers. All of them are with the NOAA Polar Ecosystems Program in Seattle. So they are on a different time zone tonight. They are coming to us after work hours. We really appreciate that. We really do. And we've got to start us off on 2024 results and 2025 plans for ice seal research in the Bering Sea and Bering Strait region, or I guess I should say Bering Strait region and beyond. We are going to start off with Michael Cameron. He is I would say you're the senior biologist, but I think your new title is you're the leader of the
Yes.
Polar Ecosystems Program. And I should say the Polar Ecosystems Program, they are the research arm for NOAA, which in this case, in this scenario, is they have authorization for research over the seals, all our seals in the state, all our whales, and sea lions. And for the purposes of this, the Polar Ecosystem Program deals with the seals, and tonight they're dealing with the ice seals. Then we will hear from Heather Ziel, and then Erin Moreland will bring us all home, and she'll give us the big finale, and we'll have questions after. Does that work for you guys? The questions after? All right, should have asked that beforehand. With that, I'm going to hand it over to Michael Cameron, the lead of NOAA's Polar Ecosystems Program. And take it away, Mike. We're so glad you're here.
Thanks so much, Gay. This is maybe our 7th or 8th time presenting on Straight Science. We really like the opportunity to come on board and talk to everybody. That was a great introduction. I'm going to repeat a bunch of that just for completeness. My name is Michael Cameron, and as I mentioned, I'm the leader of the Polar Ecosystems Program. We are part of what's called the Marine Mammal Laboratory, which is part of the Alaska Fisheries Science Center based out of Seattle. And as Gay mentioned, that's all part of NOAA Fisheries. Today, in addition to me, we're going to be hearing from Heather Ziel, who will be talking about some of our vessel-based research that we conducted earlier this year aboard a charter vessel in the Bering Sea. And we'll finish up with Erin Moreland, who's the lead for our aerial survey projects. And she's going to be talking about a large program of research we have planned for 2025. Really excited to talk to you about our big program of research. I'm just going to give you a brief overview of our program. Next slide, please, Heather. So as Gay mentioned, the Polar Ecosystems Program, we monitor and study phosid seals, or true seals in Alaska waters. And what that basically means are the ringed, bearded, ribbon, and spotted seal, which collectively are known as ice-associated seals. And we also study harbor seals, of which there are currently 12 stocks. And all of that is research that we conduct in Alaska waters. Next slide. Thanks. Just a little bit of context for the reason that we conduct our research. As I mentioned, Alaska Fisheries Science Center, there is the AFSC. We have evidence from some of the research we've conducted in the past and looking at research that others have conducted in the area that seals are responding to changes in the Bering Sea. We know that there is a reduction in seasonal sea ice, and as you might expect, ice-associated seals depend upon sea ice for some of their critical life history habitats. We've identified declines in body condition of some pups and some adults of ice-associated seals. And so it's more important than ever right now to be conducting this monitoring. There's a lot of things that we monitor that really can only be measured in the spring. And we recognize that this is the same time of year that local residents like to hunt marine mammals and conduct other subsistence activities. I like to think that we've shown over the decades of doing this work with cooperation and working with different communities that we can do this work without impacting the subsistence hunt. We have a mandate under the Marine Mammal Protection Act and the Endangered Species Act to manage and conserve these marine mammals as part of their ecosystems. But we also work together in a co-management framework with various Alaska Native groups to co-manage these species directly. Next page. So NOAA actually has a co-management agreement with the Alaska Native Ice Seal Committee. The Alaska Native Ice Seal Committee is made up of members from these 5 different regions that you can see here on the map on the left. The Ice Seal Committee also has a section called the Working Group, which includes the Alaska Department of Fish and Game, educational, and other institutions. And together, all of us work together each year to create an Alaska ice seal research plan, of which there's an example here. That link at the top actually will link to the Alaska Native Ice Seal Committee website. And from there, you can download a copy of this ice seal research plan. It's important because it shows that all of the research that we do on ice-associated seals in Alaska is presented at these ice seal committee meetings and discussed and eventually prepared as part of this research plan that gets approved by the working group. There are members from each of these 5 regions that we work with, but those members are really just conduits for communication. We do our best to try and increase our communication, increase our ability to let everybody in the region know of the different work that we're doing. We have lots of different presentations and meetings that we go to and present. And this Straight Science is just one of the many ways that we try and make sure that the communities of these coastal regions know the work that we're planning to do before we do it. Next slide. So our work can basically be divided into a few different main groups. Basically, we monitor and study seals. We conduct research on them. The research mostly is related to abundance, where they go and how they use their habitat, how healthy they are, and more importantly lately, how they're responding to changes in their environment. We do this mostly through various surveys, whether they're aerial surveys, like Erin Moreland's going to be talking about later on, or the vessel or land-based projects that Heather Ziel will be talking about. We've been doing this work for decades and have a lot of experience working in the area. We also have a very strong statistical group, and we develop a lot of new methods and equipment specifically for surveying, estimating population sizes and trends, etc. Next slide. So that's the end of my presentation. I'm now going to hand it over to Heather Ziel, who's going to give a little bit of a background on the work that she was the chief scientist for in the Bering Sea in 2024. Thanks.
Thanks, Mike. So my name's Heather Ziel, Mike just said, and I'm going to just give a brief overview about the ice seal research cruise that their program conducted in the Bering Sea this past April and May. So our goal on this cruise, as in the past, was to go to the edge of the pack ice, to sample and tag ice tag seals on the ice to collect data for several different studies. So in the past, these research cruises have been on NOAA vessels, but for the first time this year, we were on a charter vessel, the Norseman 2. We left Dutch on April 15th and traveled to the ice edge to search for seals, then returned to Dutch on May 26th. So in total, it was a 42-day cruise. So this is a track line of our cruise from April 15th to May 26th. And the ice imagery in this map is from May 6th to 7th, which was about halfway through the cruise. And the straight lines show when we were transiting directly to and from the ice from Dutch. So we can see that we spent all of our time west of St. Matthew. We started out west at the ice edge near the date line. And then as the ice retreated, we moved east to follow the edge of the ice and good habitat and the seals. So we were on the Norseman, and the Norseman's an excellent research platform. The crew is extremely experienced working in the ice. So they were able to take us up to really great ice and habitat to search for the seals. So our primary goal on the cruise was to capture seals to collect data for a variety of studies. And in total, we captured 25 seals, 11 ribbon and 14 spotted. And this table has a breakdown of the number of the age classes and sexes of each species that we captured. So we deployed tags on 20 seals total, 9 ribbon, 11 spotted. And I think I was talking about the 2 types of tags that we have. So we have the splash GPS that are glued to the hair and fall off when they molt, and then the spot tags that are attached to the webbing of the rear flipper. And then we only attach splash GPS tags to animals that were already molted so that we could get a longer retention on the animal and get more information. And then we only attach spot tags to subadult and adult animals. So we didn't attach the spot tags to young of the year and pup flippers. But if the animal was molted and old enough, then we could attach both types of tags to the animal. So 4 ribbon seals and 4 spotted seals each got 2 tags. So we get a wealth of information from the satellite tags. For example, information about location and movements, haul out behavior, and dive behavior. This map shows the locations and movements of all the seals that were tagged on this trip. The ribbon seals in orange on the left and the spotted seals in purple on the right. All of the seals were tagged west of St. Matthew. And these maps, so the points on these maps indicate the actual locations from the satellite tags. So you can see that for the ribbon seals in the orange, a couple of them went pretty far south off the shelf break. You can see this one went as far south, well, down into the North Pacific, south of the Aleutians. And this animal is still, its tag is still transmitting. And the most recent location that we got from it was from yesterday. And then you can see in the purple, the spotted seals, they mostly have stayed on the shelf. And then there's this one animal that went up north, up to near Wainwright. So we also get data about diving behavior from the tags, and these figures show the depths to which seals are diving. So the top figure shows dives from a subadult ribbon seal. And you can see it was regularly diving to between 400 and 500 meters deep. So it was, from the ribbon seal, we were seeing regular deep diving off of the shelf, which is what we would expect from ribbon seals. And the bottom figure shows dives from a subadult spotted seal. You can see it's diving to much shallower depths than the ribbon seal. Its deepest dives were a little over 125 meters deep. So also, as expected for spotted seals, we were seeing shallower dives that were on the shelf. These tags also record temperature profiles during the dives, so we can get information about the oceanography and types of water where the seals are foraging. This figure shows the dive profile of a subadult female spotted seal from late April to late May as she moved from east to west along the shelf break, heading towards the Russian coast. So the figure shows the depths and temperatures at depth for each dive. And you can see that the water temperature tends to increase as depth increases, even when diving to over 300 meters deep. So in addition to attaching and deploying satellite tags, we also collected biological samples from all of the seals. So this table has a list of the sample types that we collected and analyses that we intend to use the samples for. So we collected blood, which we will use for hematology, serology, stable isotopes, and contaminants analysis. Skin snips will be used for genetics. Whiskers will be used for stable isotope analysis. Hair will be used for stable isotopes and contaminants analysis. Nasal swabs and rectal swabs will be used to look for presence of pathogens. And scat will be used to look for presence of harmful algal blooms, and then will be processed for food habits. We also conducted other sampling on the cruise, and this sampling included, we took ultrasound measurements of blubber depth from all of the animals at 4 different points on the body. We used UAS to collect imagery of the seals. So we flew the UAS directly over the seals and pointed the camera straight down and took images. And then we took morphometrics for all of the seals. So these 3 data types will all be used to help with assessing and evaluating body condition. And then throughout the cruise, we also collected seawater. And we're working with collaborators at Bay Lab in Juneau, who will be analyzing the seawater for eDNA, so that we can try to, we'll see if we can find out any information about prey species that the seals might be consuming. So we actually already have some preliminary results from our collaborators who have finished some of the analyses. So we used PCR to test for presence of morbillivirus and influenza A, and all of the seals were negative for these 2 pathogens. Serology tests found that all seals were negative for exposure to Sarcocystis neurona. 1 out of 25 seals was seropositive for Toxoplasma gondii, and 3 out of 25 were seropositive for influenza A with the serology test. And then the serology tests found that 7 out of 25 of the seals were seropositive for brucella. And then since the satellite tags are still transmitting, and I mean, we're getting data as recently as yesterday, we're waiting on doing any analyses with the tag data. So overall, it was a very successful cruise. We captured 25 animals, deployed satellite tags on 20 of them, collected biological samples that are being used for a bunch of analyses, and conducted many UAS flights to gather information for body condition, which was a lot more imagery than we had before. And in addition to the results that we've already gotten for some of the analyses, we're still processing other samples from the cruise, and we are still getting data from the satellite tags. So we'll continue to provide updates about the data from this trip as we can. And so this is just a page with some links to web pages or sites that have more information about the cruise. So the first link goes to a page that has photos from the cruise. And then the next 3 are go to blogs that were written by Kyle Kolda. He was part of our team on the cruise, and he is a grad student at University of Alaska Anchorage right now. And then the last link goes to a page with general information about ice seal research that the Polar Ecosystem Program is working on. So after Erin's finished talking, I will paste these into the chat so that people can check them out if they want. And that's all I have. So thank you. And I can stop sharing and turn it over to Erin.
All right, I think I have to share my entire screen. So we're going to do the tunnel thing for a minute. And I'm going to try to switch it over really quickly.
Perfect.
Okay, is that working? Can you see the presentation?
Perfectly. Well
Oh,
done.
good. Okay. I can't see anybody, so go ahead and holler at me if I need to adjust anything.
We're seeing your second slide.
Okay, that's fine.
Okay.
Yeah, I didn't feel the need for another title slide. Okay, so I'm gonna share our ice seal aerial survey approach. I'm gonna give you a little overview of how we go about these surveys in general. And then for those who are aware that we were in Nome last June for a couple of weeks, give a little update on what we were doing there, and then let you know what our plans are for 2025. So the goal of our aerial surveys is to determine the abundance and distribution of ice seals. And we've been working with Fish and Wildlife and USGS to include polar bears in those surveys. We're flying many, many miles out to sea. These surveys are all really huge efforts. A lot of work goes into it. We're collecting a ton of imagery, and so the more data we can extract from the effort, the better. So we include polar bears. We've also picked up foxes out on the ice and caribou. So we try to share anything interesting we find with ADF&G and any appropriate agency or interested party. So we started all of this in 2012 and 13 with our first really large-scale surveys of the Bering Sea. And then a couple of years later, went up to the Chukchi, and then delays due to COVID landed us in the Southern Beaufort Sea in 2021. So it's taken us a decade really to survey this entire area once. This next year, we're going to try to do it all in one shot. So that's going to be great for data and just logistically, a little bit more challenging, but we're excited to take it on. So our surveys take place in the spring, which is when the seals are up on the spring ice to pup and molt. So that takes them out of the water, or many of them out of the water, and available for us to see when we're flying over. These surveys are all instrument-based, which allow us to fly at higher altitudes and faster than traditional observer-based surveys, which is where we started with all of this back in 2006 or 8 or 7, back a while ago. In preparation for these surveys, we have discussions, as Mike talked about, with the Ice Seal Committee. We make an effort to reach out to coastal communities, whaling captain associations, co-management councils to ensure that we're communicating what our plans are, and also to follow up with groups to one, let people know kind of what we're finding out, just as Heather just did, as well as to see how well what we're seeing aligns with what folks who are living in these areas are seeing. Okay, so these surveys take place using generally twin prop light aircraft. We're usually flying in the NOAA Twin Otter, which is in this picture here at the bottom, or more recently in the NOAA King Air. We've also chartered some King Air aircraft. The the aircraft requirements are that they're really long range, so they can fly a few hundred miles offshore and come back. And they need to have a hole in the floor of the plane for us to mount our cameras. So we mount a suite of 9 cameras, 3 thermal infrared cameras, 3 color cameras, high resolution, and 3 ultraviolet cameras. And they are all mounted so that we are getting these 3 strips of overlapping types of imagery, as you can see in the diagram in the lower right. We fly at an altitude of 1,000 to 2,000 feet, and then our survey speed depends on the plane. Primarily, we're usually in the 120 to 150 knot range. And then our flight lines are 25 to 50 kilometers apart, which is about 15 to 30 miles. And we collect imagery continuously while we're over sea ice. So here's an example of what the color and the thermal look like. I'll show you some UV imagery in a moment, but the UV is experimental at this point. We rely primarily on the thermal imagery for detection, and you can see why. Your eyes probably already matched up the different ice flow shapes between the two, and you've likely identified those 2 hot spots. You can match them to the seals in the color image, and then we can zoom in and see that those are bearded seals. And here's an example of a color image taken from one of our surveys that has a polar bear in it. And you can see, based on the complexity of the ice structure, it would be really difficult to see a polar bear in this kind of environment. And there he is right there in the middle. And then in the panel below, you can see how the thermal and the UV imagery can kind of help provide contrast between that animal and the surrounding sea ice. You might be noticing, in comparison to the thermal image we just looked at of the bearded seals, that there isn't quite as much contrast for this, the hotspot in this center panel that this bear is emitting compared to the background. And this is one of the reasons we've added the UV cameras to explore ways to kind of stabilize that that detection. So bears just have a much more variable hotspot signature, and we're hoping that the UV kind of gives us another angle there to help with polar bear detection. So, as I mentioned, the hotspot detection happens in the thermal imagery, and then we manually classify the species in the color imagery. And when we started out doing this in the Bering Sea, we were looking at the thermal data to find temperature spikes to figure out which frames would have seals. And we've used some kind of off-the-shelf hotspot detection algorithms. And more recently, we've developed some machine learning models to help find the hotspots, and then also to find the species in the color imagery. We don't yet rely on the color detection. We still check every single hotspot that is detected by whatever method we're using. So because we now have these color models also, there's a few different ways that we can try to integrate those into our process. So our system on the plane can allow us to, based on a detection in the thermal image, apply the color model to the entirety of the paired color image, which might result in some extra detections that aren't putting off thermal hotspots. We can also do this, what we call a late fusion approach, where the detection in the thermal image triggers an analysis of just the associated region, the appropriate region of the color imagery, to see what was there or causing that hotspot. And then there's this early fusion approach, which basically involves pixel mapping all of the appropriate imagery together so that an algorithm can can train on and then also use all of the data about that that seal or that location in the frame to help detect animals. And what we've been testing most recently is this late fusion approach, primarily because the full frame trigger increases the amount of false positive detections that need to be reviewed. And then the early fusion is probably would get us the best detections, but it's also really computationally intensive. So there's a lot of cost associated with that as far as time and computing power. And it also requires really precise alignment of the cameras and the imagery. So we've been focusing on this kind of middle of the road approach where we are getting the benefit. Oh, sorry.
Turn up with the food.
Hi, Susan, this is Gay. You're unmuted. Thank you.
Okay, thanks. So we've been kind of focusing on this middle of the road approach where we get to reduce the amount of false positives and also evaluate whether we can have models reduce the amount of false positives basically that we get from the thermal detection by analyzing the appropriate portion of the color image. Another thing our system can do is run these models in real time. And so we can, at the end of a flight, land with our detection data. Whether or not we run models in flight or post-process them, we end up with mapped areas that we covered with the camera, as well as mapped detections. So we know where the seals are that we found, and we know what area we surveyed during that flight. So when you look at these, if you see these, you see there's a number following the species name. That number is a score that the machine learning model applies to that detection, which is basically saying how well it thinks that object that it detected in that picture aligns with what it was trained to detect. So you can see right in the middle under in-flight, there's a ring seal with a 0.89 score. And then right below it, there's a ring seal with a 0.38 score. So theoretically, the ring seal with the 0.89 score is more likely to be a ring seal than the one with the lower score. But what the reality is that ring seals will get scores kind of all over the place, and we don't get a really clear definition of, above this threshold, we know it's going to be a seal, and below it, we know it's going to be a false positive. So that's some of the information we get from the detection model. So as I mentioned, we review every hotspot. So we look at each hotspot. We're able to see both the images, what is causing that hotspot in the color and the UV imagery, and then we basically go through and validate the detection as being from an animal or not being from an animal, and then in order to determine how accurate those results are, like, are we, of the animals that are that we're flying over, how many are we detecting compared to how many that are truly there? And so to get at that, we do an independent review of a subset of the color imagery. So somebody who is unfamiliar with this entire process generally looks through just a subset of color imagery, and it's not a small subset. It's like 20, it's generally around 20,000 images or more. And they look for animals and annotate them. So they draw little boxes around them. So we have, we know where those are. And then we compare our results to what they found. And it's not it's more that those animals that they found provide like a quiz for our model. So it's a way for us to test how our model's performing with the data set that we've applied it to. And we generally, from the various different approaches that we've used to detect the hotspots of animals, we find between 94 and 97% of those established known seals. So this process has worked really well over time. So that gives us our animal counts that we can map, as I showed. And then we need to get to this abundance and distribution. So we provide those counts to our statistician, and he has developed, this is Paul Kahn, has developed a spatially and temporally explicit model for seal density. And he does this using information about sea ice, snow, weather, and other covariates. And then we, within that process, account for availability, which includes, you know, how many animals are actually out of the water, which we get from the tag data that Heather was sharing. And so that tells us how many animals we're not able to account for, we're not going to be able to see because they're underwater when we fly over, which is an important element to include. Those detection rates that I just described, that how we test how well the model is performing, or how well our process of finding animals is performing for every survey. And then we also do some analyses to determine if biologists are misclassifying different species. Primarily, it's generally going to be due to the image quality, which can be really variable over the course of a survey. You can have really great images where you can tell exactly what species you're looking at, and then others that are more blob-like, which actually the last slide was a good example of. And then we also measure animal response to the aircraft itself, because if we are disturbing animals ahead of the airplane, then that is going to impact how many we can count. So we incorporate all of that into the abundance model. And then all of the uncertainty that comes from estimating these various criteria that we need to establish. And then for this example with the Chukchi Sea, we also had international partners. So our Russian colleagues were surveying in Russian waters. So that's another part of that process. And then we end up with abundance estimates for the whole area or any subset of it, and these distribution maps, which are created, or we can create, for all of the various sea ice conditions that we encountered during that survey season. So we tend to, you know, as we're surveying, the habitat is not only melting through that survey window, it's also shifting and moving. So this accounts for all of that. And you can see then how distribution might adjust due to, depending on that distribution of sea ice. So that's our general process in preparation for our surveys coming up this next spring. We were in Nome for just a couple weeks at the beginning of June this year, and we flew let's see, I think we flew on we were able to fly 9 days, and we collected over 90,000 sets of imagery. And we were doing a couple different things. We have upgraded our color cameras, which actually required some relatively major modifications to the software that controls those cameras and allows for that onboard processing of imagery, because the images are so big now. So we were testing all of that and making sure everything was working properly. We were also using a new to us plane. And there were a few elements that we needed to kind of figure out. So we were using a pressurized aircraft, which when we fly with the Twin Otters, those are unpressurized planes. You know, we don't take them up to high altitudes. We might go up to like 3,000 or 5,000 feet, or maybe as high as 10,000 feet. But that's going to be the limit unless you have oxygen. So this King airplane is pressurized, and so everything, all of our equipment is under a pressure dome to allow the airplane to have an open air hole in it with our cameras, but also to maintain the benefits of a pressurized aircraft, which we were exploring. What are those benefits going to be? How is this plane going to help us reach these really remote areas in the Bering Sea in particular, and also for the pilots to learn how do we fly this plane in the best way to get the data that is needed by this project. So it was really great to spend a couple weeks in Nome with our aircraft team and explore these challenges, and then and we were able to find kind of the sweet spot where having a plane that can go a lot faster and in a pressurized mode means that there's going to be more force put on our equipment that is exposed inside that pressure dome. So we were figuring out like how fast can we fly this really fast plane without breaking anything, and can we pressurize and fly really fast and high to, you know, south of St. Lawrence Island and then drop down and survey. So we were exploring those things and also collecting a bunch of imagery to take these color models that we've developed and have tested now. They need to be sort of refreshed with imagery from this other type of camera. So I'm sorry, I'm going to have to get up for one brief moment. I'm sorry, 1 second. Thank you. Thanks for your patience. Okay, so those images have actually gone into this new model development, and we're in the process of testing how well those models are performing right now. And I'll give you just an example of kind of what the difference in image quality is. So this is one of the bearded seals I showed you earlier. And from our old cameras and the new cameras, you can actually like see the eyes and just a lot more detail. So for our surveys for next spring, we are going to be flying, we'll have our aircraft from April 6th until June 15th. And as I mentioned at the beginning, we're going to be surveying the Bering, Chukchi, and Southern Beaufort all together. We intend to fly out of Bethel, Dillingham, St. Paul, Nome, Kotzebue, Utqiagvik, and Dead Horse, and then refueling in Inuvik in Canada. We plan the flights the night before using the most current satellite sea ice imagery. And then we send out those plans every day, which we did as well during our the couple weeks we were in Nome in June. Our flight lines extend out from shore 1 to 300 miles, and then we return to either the same airport or to another airport. And so the way it should look is these flight lines will be spread out over space and time. So on any given day, there might be 2 flights anywhere on this map in US water. And then they may kind of move around a little bit. So flights are not going to occur on days with low fog in a particular area. There's required pilot rest days, and then there's days when aircraft maintenance is required, which can take a week or 2. So we're going to be covering the area as best we can within these constraints. So we've implemented a number of mitigation strategies to reduce our impact to any subsistence hunting activities that might be going on. So just the shifting to cameras alone has allowed us to fly higher and faster, which just in and of itself minimizes our impact. Our pilots are instructed to turn around if they see any people out on the ice or in small boats ahead of the plane. And they're also instructed not to fly over any large groups of walrus or eiders, which is per our Fish and Wildlife permit, to avoid causing any kind of stampede. The pilots monitor marine radio frequencies. And as I mentioned, our daily flight plans are distributed to people throughout the region. If you're interested in getting those updates and you don't, please reach out to us and we can ensure that you're added to that list. And that email includes a phone line that goes directly to the flight team. So if there's any kind of problem with a flight plan, there's a way to communicate that. And then typically we've maintained these 30-mile buffers around communities that are engaged in subsistence hunting activities at the same time as our surveys. But these seem to be causing a little bit more problems than anticipated. Communities might be located in areas that are particularly important to include in the survey because that might be an area that animals are frequenting more regularly than other coastal areas that we're able to survey. It would only take about 30 minutes to fly to survey one of these buffers. And so we've been talking with communities about whether or not they want those buffers to be maintained. And the Bay Area Whaling Captains Association has approved dissolving the Utqiagvik buffer because they find these other mitigation efforts to be adequate. So here's an example of the type of email we send out, and then you can see a map of where we intend to fly, when we plan to be flying. We have the tail number of the airplane, and then the phone number is in the bottom and other contact information. This is just a list of this is mostly Mike's doing here. He's really put in a big push this last year to get out to a lot of these communities, and we'll continue that as much as we're able to. And then some of our communication plan to increase the use of flyers and local radio stations, and engaging in community meetings, school visits, as we're able to, either during or after the survey effort. And we're also working towards incorporating Native Alaskan participation in these surveys. That's a little bit complicated by the aircraft safety required trainings. But I wanted to make sure I mentioned it so that we can ensure that anybody who's interested, we can figure out a way to ensure that we involve people as we're able to. And that's all I have for tonight. I feel like I went longer than or it's a little bit later. I don't know if we have a lot of time for questions, but I am happy to answer any.
All right. Well, thank you all, Erin and Mike and Heather. So before we get to the questions, this is a good time for the audience to put in the chat. And I see we do have a caller, so I'm going to be right there, caller. But for right now, this is a good time to put in the chat box some love for Erin, Heather, and Michael, working backwards there. And they had to jump off the internet. There was a dog involved, which we all love dogs, and all kinds of things. And they persevered. So thank you for taking, and they're on a time that's late in the evening for them. So they do think it's later than possible, but thank you so much. And show a little love in the chat from the audience. And we'll get to questions. Now, right off the bat, we did have a first question. First off, the caller. If the caller at any time you have a question, it is just tough to be a caller on a Zoom call. So just unmute yourself and blurt out your question at any time and we'll figure it out. These guys will be happy to answer. So I know it's tough to be a caller on a Zoom call. And we thank you for being here. So the first question was in the chat and it was for Heather. And it was, can you comment on typical dive durations in minutes for the ribbon seals diving to 500 meters? Now, I saw you kind of answered it, but that other people might not have seen your answer. And it would be awesome to go back to that slide that had those,
Okay, yes.
that
Let me
diving
see.
slide that had the durations on there, because you sort of astounded me. 2 hours down. You're getting lots of nice kudos. Street Science, always a nice audience. Have to say, the best.
Am I sharing the right? Oh, hang on.
I think it's, take your time. No rush. It's only 7:30 for us. You're in the middle of the night, but we're still on. We're all good. There you go.
Does that work? Okay.
I think that must be the one. And this was from, I believe this was a gentleman named Gary.
So these figures are from a site that Josh London set up. And so I got that information from the figure that he set up. So each bar represents 2 hours binned at 5-meter depth increments for these figures.
So are you saying, I'm not sure how to read this, I guess.
So
Each bar represents 2 hours.
oops. So I think each bar, can you see my
Yep,
cursor?
I can see your cursor, yeah.
So I think each of these bars represents 2 hours, is how I understand it.
Yeah, so I suspect that what this is each bar represents a 2-hour period, and the depth indicated is the maximum depth made during that 2-hour period. It does not mean that the animal spent 2 hours at 500 meters.
Thank you.
Right.
I think the question, Heather, was when an animal goes to 500 meters, how long are those dives?
I do not actually know that from these figures.
That's good fodder for the next Straight Science when you guys come back before your survey.
I'm looking at his site to see. So he's got.
All
It's
right,
been,
well, you're
it's
working
been a
on
while
that.
since I've looked at these data, but it's on the order of, it's definitely on the order of minutes, in the 10-minute, maybe 12-minute range, sort of a thing at maximum is my memory, but it's been a while since I've looked at these data for that reason.
Good, thank you. It was Gary here. That was what I wondered. How long they were down at 500 meters and up again.
That's all right. They're going to work on that, Gary, and we'll get that answer to you. And while Heather's working on that, Peter Carey, and it's a good question, happens all the time to us, and it is tough now, but Peter Carey says, can you say something about and this might be for Mike, can you say something about the Russian border and how that complicates your survey work? And I would add, Peter, especially since we share the water and the same stocks of marine resources.
That is a great question, and it's a problem that we've encountered with this most recent set of aerial surveys. As Erin mentioned, in our first set of aerial surveys of the Bering Sea in 2012 and 2013, and when we surveyed the Chukchi Sea, we were able to coordinate these efforts with our Russian colleagues. Our Russian colleagues conducted nearly identical type surveys on their side of the line, also using thermal and infrared and visual cameras that were paired on long survey transects. We worked together with them to develop the methods, to develop the equipment, to develop the statistical models that were used to do the analysis. That was a huge benefit of the work that we were able to do earlier on in 2012 and 2013 and in 2016 in the Bering and the Chukchi. We had plans. We had already had plans in place to start working with the same set of Russian colleagues for this set of aerial surveys that were originally planned in 2024, 2025, and later. But then the Ukraine war happened, and the United States cut off our ability to communicate. We're United States employees. We cannot communicate with our Russian counterparts anymore, so we cannot coordinate surveys. I have no information that they are conducting similar surveys on their side of the border at the same time. I doubt that they are. And so we have still decided to go ahead and survey our side of the international border. So on the US side of those waters, it will make it more challenging to get an overall population estimate because we'll have to be making some assumptions, which means that our confidence intervals are necessarily going to be wider as we extrapolate the data that we collect from the 2025 survey into being able to make estimates for what might be on the Russian side. It's just a reality, an unfortunate reality we had after a long time planning and our hopes of being able to make this work, to be able to make this change, but we have to continue on and get these estimates, at least for the US side.
This is Peter. Thank you very much. That's helpful to know.
Mm-hmm.
And I would just add, I have a question on that. So on the maps you showed, when you gave the numbers for bearded and ringed, you were showing that was combined with the Russian. So are you active? That was this year? So in some way you're getting information across. You don't have to tell us how, but
No.
Go ahead, Erin.
That was from 2016. So that was the last time we had a combined effort. Yeah, I was going to add to it just that we saw a dip in the 2012 and 13 Bering Sea surveys that were collaborative with Russian colleagues. We saw a dip in ribbon seals in particular, I believe, between those 2 years. And, without without, if we, when we're in this situation where we don't know what's happening on the other side of the border, it just means, I think, that we have to be much more careful in how we interpret our results. We were thinking, okay, given this and not having the Russian data to consider yet, they could just be across the line, it could be a shift rather than a dip. And without that information, it's very difficult to know that. So I think I'm really grateful that we were able to have that collaboration for as long as we were, and hopefully we'll be able to get back to that at some point.
So what I saw was you had numbers for, and maybe this is the old stuff then, it said bearded seal. I said, don't cite. I see the gnome nuggets on, don't cite. But it said bearded seal, 147,000. That's Northern Bering Sea total 2016, or was that Northern Bering Sea only and Chukchi?
That was Chukchi only.
Okay.
And that was
This
for
year.
for 2016.
Oh, for 2016.
The Chukchi, yeah, and that's being published right now, so that'll be citable once it gets peer reviewed.
And then you had like 500,000, give or take, for ringed, but you didn't give anything for spotted and ribbon.
Yeah, and that is because we did not encounter spotted and ribbon in our survey, and that is because their distribution during that time their densities in the Chukchi are low enough that we aren't necessarily going to encounter them for that survey. So we're not saying there aren't any, but we can't say how many based on that. That's another, I'm glad you brought that up though, because that is one of the big benefits of this camera upgrade that we just did. We have a lot of, we always have a significant amount of unknown species from our older cameras. And it's, spotted seals are so kind of generic that I always felt like, there's definitely some of those are going to be spotted seals, but we don't have the image resolution to help with that all the time. So with these newer cameras, I think we'll find that we can identify ribbon and spotted seals more easily.
Awesome. And I guess that makes me think, so we don't have any numbers yet on what you found in 2024 on your survey.
Well, so our survey in 2024 was not an abundant survey because we didn't get access to the plane until June. Okay. So at that point, we did not have enough time to complete a survey, but also we were outside of our survey window. So right. At that time, you've got, all the ice is mostly gone and shifted north. There, we did notice really high densities of, oh my goodness. We have a kitten. We have a new kitten. So
that's good stuff. Nah, keep going. Don't worry about it.
She's having a ball. Let's see. We did notice really high numbers of and really high concentrations of ringed seals on the rotten fast ice in the northern Bering, and tons of bearded seals in the southern Chukchi this year. And but we're not able to do an abundance analysis on those data.
But eventually, with your flying next year, you'll be able to wrap something up there and give us a number at some point. Okay. Yes. So it's not like it's a sunk cost. And I say that because, ringed and bearded are some of the biggest subsistence and spot. Well, in this region, all the seals are important, period. Bering Strait region. But don't look behind you. Going to knock over whatever she's got going on. But the ringed and bearded are listed under the Endangered Species Act. So that's always kind of a concern for people, you know, is there some problem, or are they going up or down and all that? There seems to be a little bit more focus on those. And so you don't have a count now, but it sounds like you didn't have any kind of difficulties finding those seals in a general sense.
Right.
Yeah.
And actually this year we haven't been through all of the imagery that we collected in June, but we have seen ribbon seals in the Chukchi in those in the images that we have looked at. So I think it's still interesting seeing kind of what the conditions are at that time, and we'll have a much better picture of that this spring.
That's awesome. And we really appreciate you reporting what you could, given that you had some difficulties this go around.
Yeah.
I'll give just a tiny bit of context for
Sure.
the folks that maybe attended the Straight Science presentation, this same presentation last year. At this time last year, we planned on being able to conduct a proper survey of the Bering Sea in March, April. But as we got closer, budgetary issues and other logistical reasons meant that we weren't able to get the plane until June. It was still valuable for us to fly in June, even though we couldn't collect data useful for abundance estimating, because it allowed us to test all of our equipment. So really, the most recent work that we did in 2024 in June was just sort of a shakedown. It was just practice for the big show next year, which we're going to be using 2 aircraft covering all of the Bering, Chukchi, Beaufort, and parts of, and Bering, Chukchi, and U.S. Beaufort.
And that'll be for the big numbers. For the health of the animals, you guys were able to get in the ship, right? That was Heather.
Right.
And Heather, you could get in the ship further south. You went down, I think you guys were off St. Matthew. You were able to capture, I forget the number of all the seals you caught. And one of my questions was, I noticed on the blood test, the serology test, which you guys are taking a little bit of blood, just like you would at the doctor, and then sending that off to get tested, you know, have they had diseases? Are you guys testing for avian influenza? That has been a topic in the region. We've had avian influenza in birds. We certainly have gotten videos recently that it looks like it's still here with the seizures, repeated movements in our seabirds. We have had it here in our land animals. We know that avian influenza has jumped into marine mammals. We certainly don't want to see it in marine mammals in Alaska, but are you guys testing for that? Because we do have it both land and air and land here in the region.
Yeah, so we tested we did PCR tests for influenza A, so avian influenza. And those were all the seals were negative for the PCR tests. So those were testing for actual infection. They were testing for DNA.
Awesome.
And those were all negative. All seals were negative. The serology tests for influenza A found that 3 of the 25 seals were seropositive. So indicating that they had been exposed to avian influenza, but were not actively infected. Is what this.
Okay, that's okay. So is that a first for Alaska and waters then?
No, we've done serology tests from seals sampled between 2007 and 2010. And we did test for serology tests for influenza A, and there were some positives, not high. I forget the exact percentage, but low percentage of positive.
And do you know which brand of avian influenza? What type?
I don't know the exact, I don't know the exact brand. I can,
okay.
They were developing, they were going to potentially do some further testing. So we had this testing done working with Deb Fauquier and the people that she works with at Tufts University.
Mm-hmm.
So they were going to potentially look into seeing if they could get further testing.
I hope so. I hope so, because that is a real concern here. for this
Mhm.
region, avian influenza. And so
Yeah.
there's all kinds, right? H5N1 is the one that's the one that has jumped and has caused problems, I believe, in harbor seals on the East Coast and whatnot. So if that's something that you guys think you're seeing, I hope that's able to, Dean, I see it. I hope that's something you're able to address if a member of the public's, like all of us, are worried about.
Yeah. Yeah.
Yeah.
They
And
find out more details.
with that, Dean, go ahead. I see your hand up.
Yeah, thank you, all the speakers. I think you did a great job. In looking at health and conditioning over the years, last year you sampled 9 to 25 animals. That's a relatively small number of the total population, however they end up calculating it. Over the years, have you seen a shift in the health and condition of the populations that you're sampling? Is there a decline? Is it steady? Can you assess it at all, giving those kind of numbers of very few animals to the total population?
I'm happy to start, Heather, if you'd like, and you can chime in if you'd like. There's a number of ways to answer that question. Some of the information that we are collecting has already been published to show that there has been a decline in the body condition, and I'll explain that metric in a second. In the body condition of some of the seals that we have tested over a decadal period. I believe it is pups of harbor seals, spotted seals, and ribbon seals showed a decline in body condition. Body condition is the metric we use is the ratio of mass, how heavy the animal is, to its overall length. The idea being that length doesn't change much from year to year, but mass of an animal can change from year to year based on the environmental conditions. And because we were seeing a continual decline in the pups of those species, suggests that there was something happening that in the environment that was making it a little bit more difficult for those pups to put on the weight that they would have otherwise. The expectation then, or the assumption then, is that there's something going on that perhaps makes it just a little bit more complicated for the moms to be able to get food and turn that food into milk to feed their pups. We also found similar changes, reductions in body condition of adult ribbon seals. So you're correct that these are very small sample sizes, and the fact that we're able to detect these changes even with small sample sizes is probably important. It's also important to recognize that there's a lot bigger samples that are being collected by the Alaska Department of Fish and Game, correlated with the Alaska Native subsistence hunt. Alaska Natives cooperate with various researchers in the Alaska Department of Fish and Game to provide biological samples of the animals that they are taking. That has been going on for decades and has created a huge database able to kind of show changes in body condition of different species over time. A lot of that research actually shows that there has not been a decline in body condition of the animals that are taken in the subsistence hunt. And a big part of our research now is figuring out why there's this sort of apparent disconnect. And I say apparent because when you dive a little bit deeper into these questions, you learn that really a lot of these effects are showing up in the pups and not so much in the adult samples for exactly the reasons that I mentioned before. There's also differences in the timing of when we are out there. We're there at the time when these pups are actually being born. They still have umbilicus connected to them a number of times. So these are very young animals, and we're able to see them right at the point when they were first being born. During the harvest, of course, there are no, very little fresh newborn pups that are being taken. These are animals that are a little bit later in the season. They're a little bit older, a little bit bigger, and then of course a lot more adults than we have access to in the field. All of which is to say that it's probably safe to say there's mixed evidence, but the evidence does, on the whole, point to the possibility of ecosystem changes that could be affecting body condition of certain sections, certain segments of the various populations. It's also important to recognize that over the last decade or so, there have been two different UMEs, or unusual mortality events. Unusual mortality events are basically times in which there's been an abnormal number of strandings of animals, or an abnormal numbers of sick animals that are shown up on the beach. Over the last, I think the first was in 2011, and then another UME was declared in 2018. Those are two unusual mortality events that occurred relatively close together. And if I'm not mistaken, those are the only unusual mortality events that have been declared for Alaska seals up until that point. The period between those two was relatively minor, and they have not been able to identify the primary cause of the first UME specifically. But the second UME is believed to have been related to changes in the ecosystem, related to dramatic warming that happened in the Bering Sea in particular in 2018 and 2019. Probably going a little bit off on a tangent now from your original question, just to say that this is an area of a lot of research and a lot of interest, and something that we are very focused on, because we need to know how these animals are responding to their changing environment.
Yeah, thanks. Great job. I really appreciate that answer. It was straightforward, and I see where you're coming from. So hang in there, keep going.
All right. Thank you, Dean. Thank you, Mike. And Heather she did the job behind the scenes. She got an answer for the ribbon seal dive. Take it away, Heather. I see your answer in the chat, but not everybody's probably reading the chat, and you did it. So you should be in the spotlight for getting it done behind the scenes. Thank you.
Well, it wasn't me. I called on Josh London.
All right, Josh London.
Yes.
It's like a game show where you call in your lifeline.
Exactly.
There you
He's
go.
definitely the lifeline here, or telemetry lifeline. So he said that previous deployments, the deep dives recorded were generally 7 to 10 minutes, and up to 15 minutes for those deep dives. And for these tags that we deployed this year, I guess they did not record the dive duration for individual dives because there just wasn't enough satellite bandwidth to transmit all of the parameters. So he's gonna use the information that we get from these tags to see what is possible and what we should be collecting next time.
So then I'm just going to have to ask Mike, what is the longest dive duration of a ribbon seal, any of them, adult male? I would imagine, or female, that would probably be the biggest. Off the top of your head, don't get it down to the seconds, but.
Off the top, if I had to guess,
Yeah.
22 minutes. That's my
Okay.
guess. But I
Good
don't know
enough.
where I got that.
That's an impressive. If you're going down to, what have they gone down to? 1,200 feet in the Aleutian?
No, deeper. I think the maximums are in the 550 meter range. Actually, I'm not really sure what the absolute maximum is. They're regularly, as we've shown, going to 500 meters plus. So
okay, so 1,500 feet. That's down where you can find 1,500 feet down in the Aleutians, and that's an adult ribbon seal. Those are a high-end diver. with the chemistry and the body and everything to do that sort of thing. But thank you, because ribbon seals are kind of like just kind of awesome, honestly, but that they can do these things is amazing. That kind of breath hold and that kind of depth. So thank you for that. Any other questions? We really thank you three for taking the evening and presenting through it all. Any other questions? I do have a few announcements. Okay, next two things. One, the next Straight Science is Rick Toman from UAF Alaska Climate and Assessment Policy. We all know Rick Toman in the Bering Strait region. He takes extra care to make sure we are kind of looked over regarding our weather. He is going to be our next Straight Science speaker. That will be on the 24th, so a week from tomorrow. He is going to be here in person, so he will be here at UAF. We also will be putting it on Zoom so you can attend. He's going to give the review of what's happened to us over our summer, spring and summer, which was exciting and cold. And then he's going to give us what best predictions are for this fall and winter ahead. So that will be on the 24th at 6:30, a week from tomorrow. So don't miss that one. The other news is, and I don't know if Rick had something to do with this, but we have a enormous low about moving in, and they're taking it very seriously. So right now they're already starting to put the warnings out. It's going to come in on the weekend, it looks like, but it's coming in at 65 knots. So, or 65 miles an hour. So that's what we're expected to receive here and all the way up the coast. It's a big one, physically big, if you go to windy.com. And just putting a shout out, that'll be like a serious winter storm for us. And just take that in mind as you're planning out the weekend activities. Just keep that in mind that we have a big one rolling in. With that, we thank you three so much. If you guys would hold on and let everybody get offline just for 1 sec, that would be awesome. And with that, we'll let everyone go. We'll see you. Have a good night, everyone. See you next week. Rick Tolman.